His legs still work. His eyes still see. But he is walking into a hole in the ground behind a wasp the size of a rice grain, and he can't tell you why. He's alive, he's healthy, and the part of him that wanted to live is gone. This is Ampulex compressa, the Jewel Wasp. She doesn't poison her victim, she doesn't paralyze him. She walks her stinger into his brain, finds two specific clusters of neurons, and switches off the part of his mind that wants to live. Every other function stays online. He just no longer cares to use them. By the end of this video, you'll know how she does it. And you'll know something we got wrong about her for 140 years, until a paper in Current Biology corrected the textbooks in 2023. But first, a question with no good answer. How does a creature with a brain the size of a poppy seed know which neurons to switch off? Picture this: a garden, late afternoon. An American cockroach, 3 centimeters long, armored, 6 legs, the species that survived everything from the dinosaurs to nuclear tests. He is not an animal that worries about predators. Then a small flicker of green lands on his back, outweighed by him by a factor of 6, sometimes more. Metallic emerald body, ruby legs, Ampulex compressa. He twists, he flips, he has the strength to throw her off. He doesn't get the chance. Her stinger is already coming down, and it is not aimed at his head. It is aimed at a single specific bundle of nerves in his chest. The first sting lands in his first thoracic ganglion, the cluster of motor neurons that controls his front legs, his fight legs, his flight legs. The venom she injects there is a chemical cocktail you've heard of in pieces. 3 ingredients, 3 quiet weapons. GABA, beta-alanine, taurine. In the human brain, GABA is the molecule that helps you fall asleep. It's an inhibitor. It tells neurons to be quiet. In a cockroach's thoracic ganglion, in concentrations 1,000 times higher than anything his body normally sees, it does something simpler. It opens chloride channels. It floods his motor neurons with negative ions. The signals stop arriving. His front legs stop working. Not slowly, within seconds. But here is the thing. This is not the surgery. This is the anesthesia. The whole point of this first injection is to hold him still for what comes next, and to buy her time. The paralysis is temporary, 3 to 5 minutes. That is her surgical window. And here is where she does something almost no other animal on Earth has ever been recorded doing. She pulls her stinger back out and pushes it back in. This time through the soft membrane of his neck, slowly, carefully. And then the stinger starts to move. For up to 40 seconds, it snakes through the inside of his head. Not stabbing, not injecting, searching. Now think about that for a moment. She has no eyes inside his skull. She is feeling her way through tissue she cannot see, looking for one specific structure out of dozens. And she finds it, every single time, on the first try. So how is she doing it? In 2010, Frederick Liebersatz's lab at Ben Gurion University ran an experiment that should be in every neuroscience textbook. They took anesthetized cockroaches, opened the heads, and removed the brain entirely. Then they replaced it with a small pellet of soft agarose, a gel with roughly the right texture, but no actual neurons in it. Then they let a jewel wasp try to sting them. The wasp stung longer, much longer, sometimes for minutes, as if she could not find what she was looking for. In follow-up trials, the team varied the agarose density, softer gels, firmer gels, gels stiffer than living tissue. The stinging time changed in step. Wasps stung longest against the firmest substitutes, as if the needle was reporting back, "This is wrong, keep going." The control group, with normal cockroach brains, stung exactly once and walked away. Because her stinger was looking. The tip of it is covered in sensilla, microscopic sensory domes that detect texture, pressure, and possibly chemistry. The stinger is not a syringe, it is a tongue. She finds the brain by tasting her way to it. And what she does once she finds it is so specific and so precise that for a long time nobody believed it was real. 2 clusters out of well over 100,000 neurons. But what those 2 clusters do and what the venom does to them is where this story stops being biology and starts to feel like neuroscience fiction. Because she is not paralyzing him. She is doing something the textbooks didn't have a clean word for. The 2 clusters she targets have unwieldy names. Subesophageal ganglion, protocerebrum. Forget the words. Here is what matters. Together, they are the control centers for what neuroscientists call drive. Not movement, not muscle, the motivation to move. The internal voice that says, get up, run, fight. In invertebrates, that voice is carried mostly by a single molecule, octopamine. It is the cockroach equivalent of adrenaline, the chemistry of wanting to do something. When the wasp injects her venom into these 2 clusters, she doesn't shut his nervous system down. She doesn't damage neurons. She doesn't kill cells. She down-regulates the octopamine signal in 2 very specific places. Octopamine in the SEG, fading. Octopamine in the protocerebrum, fading. The signal that says, do something, gets quieter and quieter, and then it goes silent. His muscles still work. His sensory neurons still fire. His escape reflex circuit is still intact. He could, in theory, do anything. But there is no longer anyone inside him asking to. We have a clinical word for this in human medicine, abulia, the loss of will. It happens to people after specific kinds of brain damage. They can move, they just can't initiate. They will sit in a chair forever if no one tells them to stand up. The Jewel Wasp induces something like it, surgically, within minutes. And then she does something genuinely strange. She backs off. She lets him go. And for the next 30 minutes, exactly 30 minutes, you can time it on a watch, the cockroach grooms himself. He cleans his antennae. He cleans his legs. He cleans his face, over and over and over. It is the most thorough wash he has ever performed in his life. And it is happening because the venom contains a 4th ingredient, dopamine. Dopamine in his subesophageal ganglion triggers obsessive self-grooming. So while he stands there scrubbing himself, like a man preparing for a funeral he doesn't know he's about to attend, the wasp uses those 30 minutes to do reconnaissance. She finds, or returns to, a hole, a pre-selected burrow. Then she comes back, and she grabs one of his antennae in her mandibles. And he follows her. He follows her like a dog on a leash. A scientific paper actually uses that phrase, much like a submissive dog on a leash. She walks, he walks behind her. She tugs, he turns. He could pull free at any second. The connection is just her mouth parts on a feeler. He has the strength. He has the muscles. He does not have the will. She walks him into the burrow. She lays a single egg on his middle leg. Then she does one last thing. She trims his antenna in half with her mandibles, chewing off the upper segments, and drinks what bleeds out, a quick energy drink before the long walk home. He doesn't react. He has no opinion on his own dismemberment. She walks back out, and she seals the entrance with pebbles and bits of leaf, like a stonemason finishing a tomb. We know the off switch is real because we have found the on switch. In 2007, Libersat's team showed that if you inject an octopamine agonist back into the right region of a zombie cockroach's brain, specifically the protocerebrum, he wakes up. He starts walking again. The will returns. Which means what the wasp did was not magic, and it was not destruction. It was editing. But editing for what? Why all this? Why the surgery? Why the leash? Why the burrow? Why not just kill him? Because she needs him alive, for her larva. 3 days after she lays it, the egg hatches. A tiny grub emerges, no bigger than a comma in a sentence. She chews a small hole in his upper leg and starts drinking. Not eating, drinking. For 6 more days, she lives outside his body, sipping his hemolymph through that wound like a child with a juice box. And then, on day 9, she goes in. Now, for 140 years, this is where the textbooks told a particular kind of story. A beautiful, terrible, almost ceremonial story. The story goes back to a French naturalist named Jean-Henri Fabre in the 1880s. Fabre wrote that the parasitoid larva inside her host ate with what he called the special art of eating. She would consume the host's organs in a deliberate order, least vital first, most vital last. She would spare the heart and the breathing tubes until the very end. Because, Fabre wrote, the larva needed the host alive, so she ate him politely. She ate him like a gourmand, saving the wine for last. This story is in every documentary you've seen about parasitoid wasps. It is in textbooks. It is on Wikipedia. It has been repeated for 140 years. It is wrong. In August of 2023, a research group at the University of California, Riverside, led by Michael Adams, published a paper in the journal Current Biology. They had done something nobody had done before. They had threaded an endoscope inside a parasitized cockroach and filmed what the larva was actually doing in there. Here is why nobody had ever filmed this before. To do it, you have to thread a fiber optic scope barely wider than a human hair through the chitinous wall of a living parasitized insect, without killing the cockroach, without crushing the larva, without disturbing the chemical environment inside. The technology to do that cleanly didn't exist until very recently. For 140 years, biologists hadn't been wrong on purpose. They had been wrong because the inside of a cockroach was the most observed and least seen room on the planet. Adams's team built the first window into it. And what they saw was nothing like Fabre's elegant ceremony. The larva does not save the heart for last. She goes for it first. On day 9, when she enters the cockroach's midsection, the first thing she does is destroy the dorsal vessel, the cockroach equivalent of a heart, in the mesothorax. Average time from entry to heart destruction, 156 minutes. And then she does something nobody had ever guessed. She bites open the cockroach's tracheae, his breathing tubes, the internal airways that carry oxygen through his body. Air rushes out, into the body cavity, into the space she is living in. and she breathes it. The cockroach's lungs become her lungs. He is not just her food, he is her oxygen tank. The cockroach survives this, survives having his heart eaten, his lungs torn open because of two anatomical facts of being an insect. He has no closed circulatory system to lose pressure in. His nervous system is decentralized, distributed across his segments. He can take damage that would kill a vertebrate in an instant. So he lives, while she breathes him for several more days. But here is the question Fab never asked, and no documentary I've seen has ever answered. Why? Why the brutality? Why the speed? Why destroy the host's heart first, when going slower would keep him alive longer? The answer is this. The wasp's larva, inside that cockroach, in that burrow, she is not the predator in this story. She is the prey. Other cockroaches, Periplaneta americana, the same species as her victim, will, if they find the burrow, enter it, and they will eat her. Adult cockroaches, juvenile cockroaches, the siblings, the neighbors, the children of the very animal she is currently consuming. They are omnivores. She is meat. They do not hesitate. In a 2024 experiment funded by the U.S. National Science Foundation, researchers exposed parasitized cockroaches to hungry conspecific cockroaches at every stage of the larva's development. The numbers are stark. Day 0 through 8, the larva was killed and eaten 100% of the time. Always. Day 9, 80% killed. Day 10, 90% survive. Day 11 and beyond, she always lives. One variable explains everything: the cocoon. By day 11, she has finished spinning her cocoon, a tough silk shell that resists insect mandibles. Once it's complete, she's safe. Until then, she is the most vulnerable creature in that entire underground space. The cocoon itself is a piece of biological engineering. The larva spins it from her own silk glands, layer by layer, working inside the hollowed-out husk of the cockroach. The finished shell is rough and matte, and just a few hundred micrometers thick, thin enough to flex, tough enough that cockroach mandibles can't get a clean bite on it. They scrape, they chew, they give up. We know all of this because the team behind the National Science Foundation study did something almost cruel to find out. They opened the burrow at every single day of larval development and dropped in hungry cockroaches of the same species. They filmed the result. They timed the kills. They counted survivors. The graph they published looks like a cliff. Day 8, a flat line of 0. Day 11, a vertical jump to 100%. The cocoon, finished or not, is the difference between the larva's life and her consumption by her own dinner's family. So now the strategy makes a different kind of sense. She destroys the cockroach's heart and lungs first, not because she's cruel, not because she's polite. She does it because she is on a clock she cannot see, and the clock is counting down to the moment her cousins find her. Eating the host quickly means finishing the meal and starting the cocoon as fast as possible. Tapping into the trachea means she has oxygen even when she's burrowed deep where there's no fresh air. The whole brutal architecture of her feeding, the early heart strike, the breathing through her victim, the manic speed, none of it is dominance. It is fear. She is not the queen of this story. She is the rabbit. The cocoon is not her bed. It is her bunker. And now we can come back to the question we opened with, the question with no good answer. Her mother, the female wasp with the stinger, performed an act of surgery so precise it would shame a human neurosurgeon. She found 2 specific clusters of neurons inside an armored skull she could not see into, on the first try, every time, in a creature she had never met before, and her brain is 0.5 millimeter across. Her entire nervous system has maybe 100,000 neurons in total. How does something that small know something that specific? The answer is, she doesn't. She has no plan. She has no map. She has no understanding of cockroach neuroanatomy. Her brain doesn't know anything, but her stinger does. Every sensillum on the tip of that needle, every microscopic sensor has been shaped by tens of millions of years of selection. Wasps whose stingers couldn't find the brain didn't reproduce. The ones whose stingers could, even by a fraction more often, passed those sensors on, generation after generation for tens of millions of years. The map of the cockroach brain isn't written in her head. It is written in her needle. The cockroach in your kitchen, survivor of nuclear tests, survivor of the dinosaurs, the most successful insect lineage of the last 300 million years, has been hunted for tens of millions of those years by an animal whose injection device is smarter than she is. And the wasp herself doesn't know it. She just stings. And the stinger, after tens of millions of years of failed attempts, knows what she does not. And it has chemistry that backs the surgery. Modern analysis of Jewel Wasp venom keeps revealing new molecules, Ampulexins, tachykinins, each one shaped like a key for a single lock in the cockroach brain. The stinger finds the door. The chemistry opens it. That is the eerie, beautiful truth about her. She is not an intelligent insect. She is a vehicle. Her ancestors taught her stinger everything she will ever need to know, and her own mind has been left out of the conversation. She doesn't think about brain surgery. She just performs it perfectly, every time. If you're the kind of person who likes peeking behind the curtain of our eerie, beautiful world, the side of nature that doesn't make it into the polished documentaries because it's a little too disturbing to soundtrack with violins, then this is your channel. There is a lot of weird stuff out there. Most of it is hiding in plain sight, and I'd love to take you to the next one with me. Next time, we are going to look at a fungus that turns living cicadas into flying, infectious zombies, and gives them a working chemical addiction in the process. It is the closest thing in nature to a horror film script, and it is real. See you there.